Today, in the first session, we are going to focus on the Phenometrics, theme 1. And the first speaker is Wolfgang Busch. He comes from Salk Institute Biological Studies. He obtained his PhD from Max Planck Institute Developmental Biology, Tübingen, and then did a postdoc in Duke University. After that, he's a group leader at Mendel Institute in Vienna, Austria. And about a year and a half back, he joined the Salk Institute. He did a fantastic work in root biology. He is going to present his work on dissecting plant root growth using the high throughput phenotyping and systems genetics approach. Please welcome Wolfgang.
All right, good morning. Thanks for the kind introduction and for the invitation to Saskatoon. It's my first time here. I'm very impressed. Very nice weather. For now, I've heard. All right. So today I'm going to tell you a little bit about my work at the Salk Institute. And this is kind of a deviation from most of the other talks because I'm working on a model organism. But using the power of the model organism, we try to basically understand what are genes and variants that make roots different between different plants, and what are the molecular mechanisms that basically drive these differences, right? And so I'm going to show you a lot of little roots today. All right. So I'm really interested in one question that has been largely unaddressed despite a lot of research on model organisms, and that is what actually drives, different phenotypes between natural individuals of one species or even between different species. And the reason why this is largely not understood is that, the field has focused a lot on, artificial mutagenesis, forward genetics approaches, then basically taking these known canonical genes, expanding the knowledge, to their networks. But it's really not clear whether this is actually the same set of genes that's relevant for the phenotypic responses that we observe in natural populations. No matter if it's humans or flies or plants. And so I think just the extent of natural variation in root growth you can actually observe within the first 13 days of the life of an Arabidopsis seedling on an agar plate. As you can see here, this movie is a time-lapse over 14 days of 5 different Arabidopsis strains. They differ around in 0.3% of their genome, and still you can actually see that there's tremendous difference in root growth rate, in branching, all functional traits that over time get incorporated in the in the generation of a functional root system architecture, then presumably in the soil also, and that will have very different properties in terms of exploring the soil and absorbing and searching for nutrients and water. We are using this model system to try to figure out what are actually the genes driving this. This is something else you can study very early on already, and that's a response to environment. And we've heard a lot yesterday about the genotype by environment, and then 2 other axes even that we don't really consider so much in the basic research. And that if you put one genome, so this is one genome, so Arabidopsis is a sulfur, so all the seeds derived from one plant are isogenic. And if you change the medium a little bit, withdrawing iron or phosphate or whatever, you can actually start to see tremendous differences, right? And those are very early responses. They are most likely not because plants run out of nutrients, but it's rather more that they sense and respond to this. This is also something we can study. We can do this with many different strains and then study by the genotype by environment interaction, even in this very reductionistic system. So the way how we actually get to the interesting stuff, the genes and the variants and then the mechanisms, is that we use genome-wide association mapping. So I'm going to briefly explain it to you. So essentially, the concept is very simple. You measure a lot of phenotypes. So in this example, root growth, and then you take the genome sequence or high-resolution SNP data sets, and you try to correlate the changes, the differences in the phenotype to differences in the genotype. You do this over the whole genome. So this is the genome of Arabidopsis, 5 chromosomes, pretty small compared to many crops. And you do for every position where you have a SNP, you run a correlation do the strains that have a long root share one base, one polymorphism there, that is then different at the same position from the ones that basically have short roots. Of course, there's a lot of statistical magic involved in this. I'm pretty sure you all know about this. But essentially, that's what you do. And then you identify snips. Can I have another pointer? I think I'm running out of laser energy. So you basically try to find these peaks that basically indicate genomic loci in genes where the variation is associated and most likely responsible for some proportion of this. Okay, yeah, it's very weak, right? Yeah. It's also morning for the pointer. All right. No. Yeah, that's much better. Okay, I'm doing it two-handed, you know. All right, so the way how we do this experiment is very simple. And we actually, when I started my lab in Vienna 7 years ago, we have to come up with a solution because of a lot of solutions, at least in the Arabidopsis world, were not up to it. So we do it very simply. We grow a lot of seedlings on these plates, very tiny, don't need a large growth space footprint, but we do it under very environmentally controlled conditions. And then we basically build a very cheap scanner cluster where students can just like put the plates on these scanners, hit a button, and we basically acquire more than a couple of hundred roots within 30 seconds because this is parallelized. And then we end up with a lot of these images of seedlings on plates. Now, when I started this, I told my first PhD student, okay, so we're going to measure 50,000 roots, and now we try to do the GWAS. And there were like great papers that said, you know, there are great tools to do this. A day later, she came back, she tried out a couple of those and said that just doesn't work because there was a lot of manual correction involved in this, right? The problem is if you do it really high throughput and automated, you are dealing with, you know, every little scratch on these plates and color discoloration. So we basically, I wrote the first iteration of the software myself, where we basically used a couple of tricks back then to get a very robust segmentation of these plates. This is an example where you see the part of the plate in a time lapse, and you see what the computer actually segments as root and shoot pixels, and that actually worked quite nicely. So in fully automated roots, we actually got like to 90% of the plants that we had planted. If we didn't do any crazy conditions like stuff that colored our growth media different. So that was very good. This is basically the stuff that we have used in the past couple of years to process millions of individual images of seedlings and measure the root growth, as you can see here, but also the root topology and the root width quite accurately. And you know, we have now done a lot of studies where we then followed up, identified genes, and then dug into the molecular mechanism. So you can check that out here. Today, I'm going to tell you two stories where I think from a model system, you know, perspective, it exemplifies how far you can actually go and how this, you know, high throughput phenotyping, and I've seen like there are many more advanced methods for large fields now available here, where you can actually use this and try to get to the interesting biology. Okay, so I focus today on iron deficiency. Plants respond early on very drastically to a lack of iron in the growth media, but they do it in a genotype-specific manner. This you can see here. Those are two accessions, two strains grown on you know, happy medium, where basically all nutrients are present, and on ferrozine-containing, non-iron-containing medium, where there's no iron present. And you can see that, you know, like this accession stops its root growth like immediately, basically, while this accession actually continues growing on. So we can use these differences and conduct a GWAS, and then try to find genes and alleles. And so the first story that I tell you is telling you something about the beginning, where we used the local population here in Sweden of Arabidopsis. Just to explain this map, every dot represents an Arabidopsis strain that has been collected in the last 60 years by Arabidopsis researchers and sent to the stock center that started already in the 30th of the last century. And you can see there are thousands of strains coming from very different environments, which we can use then to study, to not only harness the phenotypic variability, but also to study the gene by environment interaction and potential adaptive value of variants. So we focused on the Swedish population because these Arabidopsis strains are pretty much related. They basically migrated into Sweden from the south after the last ice age, but then basically diverged. And there's a lot of difference between the southern and northern Swedish Arabidopsis, including a lot of phenotypic variation. All right, so using these 130-something lines, we basically found a lot of phenotypic variation for this response in iron. So you can find accessions that basically stop their growth very fast, and then accessions that continue to go longer. We use this to conduct a genome-wide association mapping, and you can see this peak on chromosome 1, on the arm of chromosome 1, basically reaching beyond our significance threshold. So that was the region that we then decided to study. So because we have this 1,000 genomes projects where we have 1,000 genomes sequenced, by a couple of labs in the Arabidopsis world, we basically can really zoom in for all these accessions and look at the individual polymorphisms in this region. So this one here was the marker SNP that we mapped, where you can see that actually all the accessions that have a long root, like these ones, compared to the accessions that have a short root, like these ones, actually have an A here, right? All the long roots have an A, and all the others have the reference allele of this polymorphism. But something that took our attention was actually this region here, where within the long roots, there are differences to the short roots. But there's actually allelic heterogeneity, so they are slightly different, like independently evolved SNPs that are marked by the same haplotype. The reason why we were immediately drawn to this area was because this gene is a very famous iron gene. It's called Fro1, that has an essential function in Arabidopsis and other plants, mostly dicots, to reduce iron in the soil to make it available for uptake. So in the soil, there's a lot of iron on the planet, but still most of it's not available because it's in a higher order redox state. So Fe3+ cannot be used and taken up by the plant. So what plants do is that they reduce the iron 3+ to iron 2+ that then can be taken up. This reduction, which is essential for this, is done by Fro2. So basically, this was an excellent candidate genes. So we did the following to really test. Like it's often if you follow candidate genes, the big danger is that you're wagging your own tail. You think something is causal in that region because there's a known function, but you cannot be sure of this, right? Because it just might be something else that hasn't been discovered. So in order to avoid this, we always do these allelic complementations where we take the mutant, and in our case, the mutant Fro2 line that doesn't have a function of Fro2 actually grows slower under iron deficient conditions. We take these alleles from, including the promoter and the coding region from Fro2, from these different 3 lines. And the hypothesis would be if we transform this transgenically into our Fro2 mutant plants, these alleles would confer a higher root growth rate upon iron deficiency, and these alleles would not complement the phenotype much. And that's exactly what we see. We do this in T1 to have a lot of different insertions to avoid confounding due to insertion site. But essentially what you can see here, that the long versions of Fro2 confer a much higher growth rate on iron deficiency than the short allele versions, right? And actually the intermediate allele is somehow in between. So that told us Fro2, allelic variation of Fro2 is truly responsible for this. We can then figure out what actually happened. In this case, it's pretty easy. There were no protein changing SNPs. They were all non-coding or in the promoter. And this is what we see here. Really expression, the main difference between these transgenic lines is that in this line with the long Fro2 version, there's more expression of Fro2 than in the other line. And this actually translates into a higher iron reductase activity because we can biochemically measure this. And basically this allele does a much better job because there's more of it to reduce the iron. Okay, so obviously, and a lot of you will ask yourself, is like this Agar plate, like very early phenotype, something that might be of relevance? And that's what we test here. We increase the alkalinity of the soil, which leads to less available iron for the plant. And essentially, we can observe that this basically long root Fro2 allele is much better in coping with this compared to the mutant or the short root allele, telling us that these small differences basically get translated into an organismal phenotype, and that may be related to local adaptation. Local adaptation is something that we can try to understand using another dimension of the data, because we know where these lines came from. Here I basically show you a map of Sweden, colored by the different soil types. Obviously, soil is very important for roots and for iron uptake. And I colored these accessions in either green or black, depending on the Fro2 allele that they have. So the fast-growing allele is this allele, the black one, and the slow-growing allele is the green one, right? And what you can immediately see that in southern Sweden, there are many more of the slow growth conferring Fro2 alleles, and in northern Sweden here, there is almost exclusively the A allele, right? And so the main difference between these two geographical areas is actually the diversity of soil types. So this here is actually pot soil. It's a highly acidic soil where iron gets washed out pretty rapidly, at least in the depth when you think of like Fro2, like early seedling establishment. And then these green guys with a slow growth Fro2 allele on iron deficiency basically are almost excluded from the pot soil, right? So an hypothesis that we only can test when we go to Sweden and plant these plants, which we haven't done, it would be that these different Fro2 alleles confer an advantage in podzolic conditions. So yeah, check out the paper if you want. But basically, the next step would be to go to Sweden and test this, or to other pot soils and see this, and that might be actually interesting for crops as well, right? It's maybe just one thing, by data analysis, it is not completely comprehensively possible to make such a conclusion, because you have population structure. So all the northern Arabidopsis are more related to themselves than the southern. So if you correct for population structure, you just go in the marginal association value. So you really need to do the field experiment. But I'm kind of scared of Sweden in the winter, so. Anyway, so what's the next step? So the next step is actually to go beyond the single locus, right? We all know that genomic prediction works because it basically guesses the whole genome, or a large part, right? So obviously, if we want to think about mechanism, we want to go beyond the single gene using the power of our phenotyping. So if we want to do this, if we want to try to identify pathways or networks that maybe are independently targeted by natural variation to confer the same phenotype, we have to think about networks. And we have to basically do a better job in getting more significant associations, right? So you have seen that for the last trait I showed you, there was one association. That's our typical, picture for 200 to 300 Arabidopsis exception. It's much better than in human, where people have to go up to 50,000 individuals to get a decent number of peaks, but still, right? So the key here is to increase the throughput. So we basically put some more manpower on this and phenotype 500 accessions. And what we get is actually a large number of associations, much more than we would expect by a linear increase, right? So basically, numbers is the thing in getting a decent number of associations to start to think about networks, right? And to start to think about interacting genes and pathways that you might be able to target more efficiently than the single gene. And maybe that gives you also a better perspective to transfer trait regulation between different species. All right, so we have a lot of SNPs, and so the next thing is we need to somehow know how genes that are close to the SNPs connected to make an inference about networks. So for this we used ARANET, which is a genome scale network that basically in Arabidopsis integrates 20 or more different data sources like protein-protein interaction screens, but also just co-expression or literature mining. And that gives you essentially the probability that any two given gene of the Arabidopsis genome might functionally interact. And so when we basically filtered candidate genes that were very close to these peaks, whether there is some evidence that they interact in this network model, we actually found a couple. And this cluster of leucine-rich repeat receptor kinases came to my attention because these type of genes are very interesting to think about responses to the environment. Mainly because previous work, mainly in Arabidopsis, has, but there are also many known cases of their role in defense responses in rice, for instance, that these leucine-rich repeat receptor kinases can actually get external signals. They have this external leucine-rich repeat domain that can be used for receptor functions, and an intracellular domain that is used for signaling functions. They can actually transduct signals from the outside to the inside. And the very interesting thing, and that was pioneered by my colleague Joanne Corey at SOG, is actually that the view is emerging that there's actually an interaction, a competing interaction between different receptor kinases, so between receptors and co-receptors that basically can respond, where you can have modules that respond to multiple cues. And this is kind of the only like really good example until now I'm going to show you, we found, where basically this module, BRY1, BAC1, FLS2 could respond to either brassionosteroid, which is an important growth hormone, or flagellin, which is a signature of pathogens. Okay, so first thing we did was to basically check whether our 4 genes here, which are 3 receptor-like leucine-rich repeat receptor kinases and 1 protein kinase, actually have a phenotype when we knock them out single, right? And you can see that actually 3 of those 4 guys show reduced root growth upon iron deficiency compared to wild type, and 1 of these guys is actually slightly increased. So we also did like higher order mutants for all of them. That took us a couple of years, even in Arabidopsis, but I'm not going to talk about the data today because I just have 30 minutes. All right, so we were thinking, so what's actually the mechanism for this iron response and these receptor kinases? And what you can see here is something that captured our attention very early on when we basically used these SIR3 receptor kinase that we had found, one of these guys, and put it into tobacco with a fluorescent protein. When we treated it with iron depletion using ferrozine, actually the signal disappeared from the membrane. You can see that in a Western blot. So essentially, when you remove the iron, that basically goes away from the membrane. The other interacting kinase, FIRE1, the other receptor kinase, stays on the membrane. So we think there's actually this interaction of these kinases on the membrane, and when there's iron deficiency, one of them goes away, the other one is free to interact with another partner in this module. So that's the hypothesis. Is that true for the root? I mean, that was a tobacco assay, so we generated stable lines. You can nicely see a great Arabidopsis root here. And you can see that the green signal, which depicts the SIR3 protein under its native promoter, is expressed in basically all the cell types and in an interesting manner, in polar manner. So when we then did the same experiment with the iron withdrawal in roots, using our long-term automated confocal pipeline that we have developed, where we basically can put multiple roots under a confocal but change their conditions individually and use a software that operates the microscope to track the roots because they grow, so you need to keep track of the root tip. You can actually see that under control conditions, when you treat a root with regular medium that has all the nutrients, basically continues to grow, and it also sustains a signal. So this violet signal is this SIR3 protein, a signal throughout its growth. But when you withdraw the iron at the same time, you basically see 2 things. One is that it basically stops growing, so no iron, it stops growing. And at the same time, this is coincided with this basic depletion of the signal that only gets stronger again when it starts continue growing. So this SIR3 receptor kinase is basically probably necessary to retain growth. So when it's gone away, there's the signal stop growth. So we did this, of course, because it's automated. We like statistics. We extracted the signal, and you can see under iron-depleted conditions, this is actually true compared to iron-sufficient conditions for a large number of fruits. So that's at least 12 repeats. Okay, so we think that's true. Something else that came up that's interesting I want to highlight here, that actually 2 of these 4 genes that we found using this GWAS network analysis were implicated in defense responses. So the SIR3, the one that disappears from the membrane, already a couple of years was found in a screen using a recombinant inbred combination line collections to be responsible for essentially autoimmune responses. So you basically, the immune system is activated without any signal present. And then we found in a large screen for genome-wide interaction of receptor kinases together with Youssef Belkadir, that actually one of our guys was in a central position interacting with FLS2, which is the flagellin receptor, a central component of pathogen-associated signals. So we tested whether iron acquisition and pathogen signaling actually kind of coincides at the point of our receptor kinases. And so we did this experiment. So we looked at the iron transporter reporter line. So this reporter, the IRT1 transporter, is never induced when you are under iron sufficient conditions, right? And we used an inactive form of flagellin to treat it, and there was no signal as expected. But then when we treated it with flagellin, with the active form of flagellin, actually plants immediately upregulated the iron acquisition strategy. I mean, it was known from a lot of descriptive data that there is a crosstalk of iron deficiency components and pathogen signaling. Also in mammalians, it's very important as a first response to infection. But that was very interesting for us. And most interesting, if we did the same assay in our receptor kinase mutant, SIF3 that goes away from the membrane, we actually found this induction is, now I got confused, sorry. This induction is actually greatly reduced. So at least part of this crosstalk between those two pathways is mediated by these receptor kinases. And it's hard to think about a mutant screen, a general like artificial mutant screen where you could have found these things, right? So I think this is the true power of natural variation and GWAS and combined with network analysis that you can actually find this. So based on other, like a lot more data, including an epistatic analysis of all these mutants and mutant combinations, we actually think that 3 of those receptor kinases interact on the membrane. They somehow compete with a pathogen related receptor that detects flagellin to determine whether there's defense or growth. And the interesting thing is that, we found these four guys in a GWAS, so we know that genetic variation actually affects their function. So we actually think this thresholding that might occur between iron and flagellin might be tuned by those two different versions. So if we truly understand how these different polymorphisms in these receptors tune whether they decide to stop growth and defend, this will actually lead to principles that we can use for modeling to tailor plants to it. Okay, so this is my last slide. I think I'm still in time. So you see, like we're talking now about combinations of conditions. And of course, yesterday there were great talks about, about soil. There's a lot of different conditions in soil. We use these plates. So we have to start to kind of like get asymptotically closer to the soil and generate conditions in which actually signals and conditions are combined at different levels. And so my postdocs were not so happy about the prospect of pipetting all day long. So we built together with Waco Lab Automation this robot we called Larry, because we got the money for it from a crowdfunding campaign, and the major donor was Larry Greenfield. And so that's what Larry does all day. It takes exactly one seed, and puts it on a very distinct position of the plate that our software knows about, so that helps for the segmentation of these images very much, and we can actually produce 5 weeks amount of work in less than 1, right? So that's kind of the next step. Then we basically found that we run into other bottlenecks. I guess other people have experienced that. You solve one problem, you open up a box of worms. So one is seed production, so Larry is hungry for seeds. We need to produce way more seeds for Arabidopsis. And then also, because we have a much larger number of images, if we're dealing with 90% sensitivity, we have a problem, because that we have to do 10% more experiments. And so we actually have developed deep BRAD, or we're currently developing it, deep vision, deep machine learning based BRAD, also using some of the approaches with convolution, deconvolution that were proposed yesterday, to basically get to 99% of sensitivity, which is actually in the large scale of things a lot, like 9 percentage points of increase. Okay, with that I'm going to close. Most of the work was done that I presented today with the iron was done by Santosh Satbhai. He moved on, got a faculty position in India last year, and the work is continued by 2 other postdocs, Matt and Chin Chin, and Han Wan is actually responsible for this deep BRAD. And with this, I thank you for your attention and take any question.